1 billion years ago, Earth would have looked almost unrecognizable. The continents were joined into a single landmass, surrounded by one enormous ocean, while the land itself was almost completely lifeless. No trees, no insects, no birds, nothing moving across the surface. Above it, the sky carried an orange-tinged haze, and the atmosphere contained oxygen at only around 2% of modern levels. There was oxygen here, technically, but not enough to make the air safe for a modern human to breathe for long. The ocean was stranger still. It was not blue. Near the surface, parts of it were milky turquoise. Deeper down, the water could become almost black, filled with chemistry unlike anything found throughout the oceans today. There were no fish, no coral reefs, no familiar marine predators. For 1 billion years, Earth appeared to barely change. Geologists eventually gave this enormous stretch of time a remarkably unimpressive name, the boring billion. But something important was happening beneath that apparent stillness. Inside this strange atmosphere and hostile ocean, one of the most consequential biological developments in Earth's history was taking place. It left almost no obvious mark on the landscape, but its consequences are inside every living human being today. And this world would eventually change again. The oceans would freeze on a scale almost impossible to imagine, locking much of the planet beneath ice for millions of years. What survived that transformation would help shape everything that came afterward. The strangest part is that the billion years that looked like nothing were quietly preparing the world for almost everything. In this story, we're going back 1 billion years to a version of Earth that looked nothing like the planet we know, to uncover what was happening beneath its strange skies and lifeless landscapes, while the foundations of complex life were quietly being built. Before we continue, if you're enjoying these journeys through Earth's strangest chapters, take a moment to like the video and consider subscribing. And let me know in the comments, where are you watching from today? Settle in and let's return to the Earth before complex life took over. The story enters a stretch of Earth history that lasted so long that calling it a chapter almost feels too small. The Mesoproterozoic and early Neoproterozoic occupy the middle portion of the Proterozoic eon, covering roughly 1.8 billion to 700 million years ago. The Proterozoic itself extends from 2.5 billion to 541 million years ago, nearly 2 billion years in all, making it the longest named eon in Earth's history. By this point, Earth had already passed through its most violent beginnings. The Hadean had brought a young planet shaped by immense heat, impacts, and a molten surface. During the Archean, the first life appeared, along with early bacteria and the first forms of photosynthesis. Then, around 2.4 billion years ago, cyanobacteria began releasing oxygen on a planetary scale during the Great Oxygenation Event. That transformation altered the atmosphere and oceans, changing the chemical direction of the planet for good. Around 1.8 billion years ago, the landscape had settled into a very different kind of world. The crust had become more stable, oceans were established, and continents had developed recognizable forms. Plate tectonics still moved the pieces of the surface, raising mountains and opening seas, although the planet no longer carried the raw appearance of its earliest youth. Earth had reached an age when its foundations were established, while its biology remained surprisingly modest. This is where the long middle chapter begins. Oxygen existed in the atmosphere, though only in limited amounts. Continents drifted across the globe, oceans covered much of the surface, and microbial communities occupied the places where conditions allowed them to flourish. To a human observer, however, the planet would have offered little evidence of the complexity that was eventually coming. The foundations were present, yet the architecture of modern life had barely begun to emerge. Reconstructing this interval is difficult because the planet has been remarkably effective at erasing its own history. Rocks are buried, heated, folded, dissolved, and recycled through tectonic processes. Sediments disappear beneath younger layers, while erosion removes landscapes that once stood above ancient seas. A record approaching 1 billion years can therefore become fragmented into scattered pieces, with enormous stretches represented by little more than a chemical trace. Those traces are where the story survives. Ratios of elements preserved in minerals can reveal how ancient oceans and atmospheres behaved. Magnetic minerals can retain the direction of Earth's magnetic field from the moment their host rocks formed, offering clues about where continents once rested. Tiny fossils preserved inside exceptionally resistant rocks provide glimpses of organisms that lived when the planet looked almost empty. Each clue is incomplete on its own. Together, they form a faint outline of a world that has almost vanished. The Proterozoic leaves no single window through which its landscapes can be viewed. Instead, scientists piece it together from scattered chemical signatures, magnetic memories, and microscopic remains, allowing an otherwise lost age to take shape again. Earth was still moving beneath its continents, exchanging material among rock, ocean, and atmosphere, while life altered its surroundings at microscopic scales. The changes unfolded across spans too immense for any generation to notice. The Proterozoic was therefore no empty interval between dramatic ages. It was a long period of connection between geology, chemistry, and life, preserved today in fragments that scientists are still learning to read. Imagine measuring the ancient atmosphere one molecule at a time, and the numbers begin to make the planet feel unfamiliar. Around 1 billion years ago, oxygen may have reached only about 2% of today's atmospheric level. Estimates across the boring billion range more broadly from a fraction of 1% to roughly 2% of modern atmospheric oxygen. For a human accustomed to present conditions, that difference would have been profound. Modern air contains about 21% oxygen. Human consciousness generally requires an oxygen concentration of at least 19.5% at normal pressure, while extremely low concentrations can cause unconsciousness within seconds. The ancient atmosphere sat nowhere near those conditions. A person arriving there without protection would have been breathing an atmosphere incapable of supporting normal human life, even though oxygen was already part of the planetary system. Other gases shaped that atmosphere as well. Methane remained present in some intervals, along with other chemically reducing gases left over from an earlier Earth. Their amounts probably changed through time, influenced by microbial activity, volcanic processes, and interactions among the atmosphere, ocean, and minerals. Methane mattered especially because it could react with sunlight high above the surface, helping create hazes that altered how sunlight moved through the atmosphere. Something else was developing in the same chemical landscape. Oxygen exposed to ultraviolet radiation can produce ozone, a gas capable of absorbing much of the sun's harmful ultraviolet energy. Modern Earth's ozone layer depends on an atmosphere containing enough oxygen for this cycle to operate efficiently. During much of the Proterozoic, oxygen remained too scarce for a strong modern style ozone shield. The distinction matters when we imagine the ancient continents. Sunlight reached the surface carrying ultraviolet radiation that today is filtered substantially before it arrives at ground level. Bare rock had no leaves, bark, soil, or other biological cover to provide additional protection. Any organism attempting to establish itself there faced intense radiation as well as drying, temperature changes, In the absence of the established nutrient cycles that later ecosystems would depend upon. Life therefore remained concentrated where water could provide a physical barrier. Shallow seas, coastal sediments, and protected environments offered places where ultraviolet radiation weakened as it passed through the water. Even there, survival depended on microscopic communities that had evolved to work within narrow chemical limits. The strange relationship between oxygen and ultraviolet light gives this atmosphere an unusual character. Oxygen was one of life's great products, released through photosynthesis for immense spans of time, yet its concentration remained too low to make the air breathable for humans. At the same time, its gradual accumulation was beginning to support the chemistry that could create ozone, while still falling short of producing the protective atmospheric layer familiar today. Earth was therefore caught between two atmospheric states. The old chemistry had not completely disappeared, while the oxygen rich world of later ages had not yet arrived. Above the continents, sunlight crossed an atmosphere still carrying the signatures of an earlier planet. At the surface, rock waited for biology to find a way onto land. The air itself marked the boundary between those two worlds, carrying just enough oxygen to transform Earth's chemistry, yet far too little to make this ancient atmosphere a place where a human could breathe directly. Look upward from a shoreline 1 billion years ago, and the first unfamiliar thing would have been the color of daylight itself. The atmosphere was transparent enough for sunlight to reach the surface, yet its chemistry differed from the mixture surrounding Earth today. The familiar blue overhead would likely have been replaced by a softer palette, shaped by gases and particles moving through an atmosphere with much less oxygen. The blue sky of the present world comes largely from Rayleigh scattering. Nitrogen and oxygen molecules scatter shorter wavelengths of sunlight more effectively than longer wavelengths, spreading blue light across the sky in every direction. That process still operated in the Proterozoic atmosphere, though the ingredients surrounding it were different. Oxygen was scarce, and methane appears to have been more important during some intervals, while tiny particles and atmospheric aerosols may also have altered the passage of sunlight. Methane can interact with sunlight in ways that create atmospheric haze. When concentrations become sufficiently high, chemical reactions driven by sunlight can produce complex carbon-rich particles that absorb and scatter incoming light. Blue wavelengths are especially affected, allowing warmer tones to become more prominent. The resulting appearance has been compared with the orange and amber haze surrounding Titan, Saturn's largest moon. Although Earth's ancient atmosphere would have followed its own chemistry and would not have been an exact copy, there was probably no single color that represented every day across the entire billion-year interval. Depending on methane abundance, atmospheric particles, humidity, and local conditions, models allow a range from pale orange to a diffuse, washed-out appearance. Some skies may have carried a muted yellow or cream quality, while others could have developed stronger reddish tones. The atmosphere itself was therefore part of the landscape, changing how distant mountains, coastlines, and clouds would have appeared beneath the sunday. Even the sunlight had a different character. 1 billion years ago, the sun was roughly 6% less luminous than it is today. Our star has gradually increased its energy output throughout its lifetime, so the ancient Earth received less solar energy despite orbiting the same star. This was part of the long-standing faint young sun problem, in which geological evidence shows that Earth remained capable of supporting liquid water even while the sun was weaker than it is now. Imagine that reduced sunlight passing through an atmosphere carrying a faint orange haze. Across the horizon, there would have been no forests to interrupt the view, and no broad canopy to divide the sky into patches of shade. The land would have presented mineral surfaces beneath a star that looked familiar in shape, yet delivered a softer amount of warmth. Clouds, when present, would still have moved through the atmosphere, reflecting and reshaping the sunlight. Water would still have gathered in seas and shallow basins, giving the planet an active cycle of evaporation, condensation, and rain. Yet the color of the air would have changed the entire visual character of those familiar processes. For a human standing there, the sky would have offered a subtle reminder that planetary appearance depends on chemistry. The same sun could illuminate the same world under a different atmospheric mixture and produce an entirely different visual impression. Earth had already become oxygenated enough to transform its chemistry, yet its atmosphere still carried enough of its older character to tint the daylight in ways no modern landscape can fully reproduce. At the surface of the Proterozoic Sea, sunlight reached a thin living layer where cyanobacteria gathered energy. A little farther down, the character of the water changed completely. The ancient ocean was divided by chemistry, with oxygenated water above and oxygen poor, sulfur-rich water occupying much of the depth below. This arrangement created a boundary between two environments that could exist together while remaining profoundly different. The idea became influential through the work of geochemist Donald Canfield, who proposed in 1998 that large portions of the deep ocean had been anoxic and euxinic. Euxinic water contains both extremely little oxygen and abundant hydrogen sulfide, a compound associated with the familiar odor of rotten eggs. In this setting, the deep sea could support microbial metabolisms that depended on sulfur chemistry while leaving little room for organisms requiring oxygen. The separation was maintained by density and circulation. Warm, illuminated surface water supported photosynthesis, while deeper water received organic material sinking from above. Microbes consumed available oxygen as that material was broken down, and the deeper environment became increasingly reducing. Hydrogen sulfide could then accumulate, especially where sulfate-reducing microorganisms transformed sulfur compounds while consuming organic matter. The upper ocean could therefore remain productive, while the deeper water accumulated a very different chemical character. Organic matter falling from sunlit layers became food for microbes, linking the surface to the depths. Each particle carried carbon and nutrients downward, feeding reactions that helped preserve the separation between oxygen-rich and sulfide-rich waters. Such water would have been deeply inhospitable to complex animals. Hydrogen sulfide interferes with cellular respiration, disrupting the molecular machinery cells used to obtain energy. An organism entering these waters would encounter a chemical environment capable of overwhelming oxygen-dependent metabolism. The deep ocean, therefore, formed an enormous biological boundary, limiting the places where more energetically demanding life could thrive. The chemistry also affected the appearance of the sea. Modern ocean color comes from the interaction of sunlight with water, suspended particles, dissolved substances, and microscopic organisms. Ancient water carrying abundant sulfur compounds and microbial material would have behaved differently. Some models of Proterozoic ocean color suggest that surface regions could have appeared pale turquoise, where finely dispersed elemental sulfur scattered incoming light. Other areas may have looked far darker. Sulfide interacting with iron and organic material could produce particles that reduced the penetration of light, giving portions of the water a deep, almost black appearance. These colors would have shifted with depth and chemistry, producing an ocean whose appearance changed across invisible boundaries that stretched through the water column. There were no fish moving through this world, no coral communities spreading across reefs, and no shells accumulating along familiar seafloors. Microbial mats occupied the productive margins, while chemical gradients created habitats for organisms adapted to sulfur, iron, and other compounds. Life was abundant in microscopic terms, even when the ocean appeared empty to human eyes. The Canfield Ocean was therefore more than an unusual body of ancient water. It represented a planetary system in which oxygen, sulfur, iron, and nutrients were continually being redistributed between surface and depth. The boundary between these layers shaped where energy could be harvested and which forms of metabolism could persist. Across immense stretches of ocean, chemistry determined the limits of biology, leaving a world whose waters carried the signatures of life without yet supporting the complex communities that would later fill the seas. A memory of the ancient ocean survives in stone, written in bands of red and gray that cross landscapes on several continents. Long before animals appeared, seawater carried enormous quantities of dissolved iron. The metal remained suspended in the water until changes in ocean chemistry allowed it to combine with oxygen and form solid minerals. Layer after layer accumulated on the seafloor, preserving a record of an ocean whose chemistry had no modern equivalent. These deposits are called banded iron formations. They consist mainly of alternating iron-rich minerals and silica-rich layers, often arranged in stripes that can extend through enormous stretches of rock. Their presence tells scientists that iron was once abundant in seawater, while oxygen was being introduced into particular parts of the ocean. Each band represents a moment in a much older chemical rhythm when dissolved materials moved through the water and eventually settled into sediment. Some of the most important iron deposits known today belong to this ancient history. The Iron Range of northern Minnesota contains enormous quantities of iron-bearing rock. Across Australia, the Pilbara region preserves formations created during the same broad chapter of planetary history. In Brazil, the Quadrilatero Ferrifero contains another major concentration. Modern landscapes therefore expose fragments of an ocean that disappeared long before human civilization began. The chemistry changed as sulfur became increasingly important in deeper waters. Sulfide reacted readily with dissolved iron, producing iron sulfide minerals such as pyrite. Once iron was captured in this form, much less remained available to combine with oxygen and settle as iron oxide. Banded iron formation became far less common around 1.8 billion years ago, close to the transition toward the chemically stratified ocean described earlier. The disappearance of these formations does not mean iron vanished from Earth. The element continued moving among rocks, water, sediments, and living organisms. Its location and chemical form changed. In one environment, iron could remain dissolved. In another, it could become locked inside a mineral and buried beneath later sediments. The ancient ocean was continually sorting elements according to the chemistry of each layer. There is something especially tangible about this record because the iron eventually became part of the human world. Ancient deposits have been mined for generations, crushed, processed, and transformed into the steel used in bridges, buildings, railways, machines, and ships. The material surrounding modern life can therefore contain atoms that once circulated through seawater long before animals existed. Their presence connects an industrial landscape with a sea floor that existed billions of years before any human hand shaped metal. Those atoms passed from seawater into sediment, from sediment into rock, and from rock into human industry. Their journey crossed changing continents, altered climates, shifting oceans, and countless generations of life. The striped formations remain in the ground as a physical memory of that journey. Their alternating colors preserve changes in chemistry that unfolded on a planet still developing its atmospheric and biological character. What looks like ordinary rock can therefore hold the trace of an ocean, its dissolved metals, its shifting chemistry, and the gradual rearrangement of elements that continue to circulate through Earth today and onward through time. Long before coastlines were divided among many continents, Earth's land gathered into a single enormous arrangement. Around 1.1 billion years ago, the supercontinent Rodinia brought much of the planet's exposed crust together, forming a world whose geography would have been difficult to recognize from any modern map. The name comes from a Russian word meaning motherland, an appropriate name for a landmass that gathered distant pieces of Earth's crust into one broad body. Rodinia was positioned roughly around the equator and surrounded by a global ocean called Mirovia. Most of the planet remained covered by water, leaving the supercontinent as a solitary expanse within an immense marine setting. Its coastlines stretched along shallow seas, while the interior reached far from the influence of those waters. The arrangement created enormous distances between the center of the landmass and the nearest ocean. Conditions deep inside Rodinia would have differed sharply from those along its margins. Rainfall arriving from the surrounding ocean would have lost much of its moisture before traveling far inland. Rivers could cross portions of the continent, yet reaching the deepest interior would have been difficult. With little biological activity on land to retain water and build soil, exposed rock would have dominated the interior, forming broad regions where mineral surfaces met an atmosphere carrying intense ultraviolet radiation. The margins offered a different setting, although they were still worlds apart from modern shorelines. There were no forests leaning over beaches, no birds moving above tidal flats, and no kelp forests beneath coastal waters. The meeting point between land and sea was shaped mainly by rock, sediment, waves, and microbial communities. Shallow water could receive minerals washed from the continent, creating productive environments for microscopic life near the edges of Rodinia. Scientists know the outline of this lost continent through a form of geological memory called paleomagnetism. When certain minerals crystallize inside cooling volcanic rocks, they can preserve the direction of Earth's magnetic field at that moment. Because the magnetic field changes with latitude, these minerals provide clues about where a piece of crust was located when the rock formed. Combined with matching rock layers, mountain belts, and ages across separated continents, those magnetic signals allow researchers to reconstruct pieces of Rodinia. Laurentia, the ancient continental block that forms much of present-day North America, occupied a central position within the reconstruction. Other pieces now belonging to Antarctica, Australia, and South America were attached around its margins. Their arrangement is inferred from geological relationships that have survived immense spans of tectonic movement, erosion, burial, and uplift. The forces that brought these pieces together also raised mountain ranges across Rodinia. Some of those belts may have approached the scale of today's Himalayas, creating highlands that began eroding almost as soon as they formed. Rain and streams carried fragments downhill, transporting sediments toward shallow coastal seas. Grain by grain, the mountains returned material to the ocean, connecting the rising interior of the supercontinent with the microbial worlds along its margins. Rodinia was therefore more than a single piece of geography. It was a planetary arrangement that changed how water, minerals, sediments, and climate interacted across Earth's surface, giving the ancient world a distinctive rhythm shaped by one continent surrounded by one global sea. Across those distances, the planet's surface remained governed by the slow movement of rock and water, with no permanent boundary between geology and climate. Across Rodinia, the ground offered little of the living surface we associate with Earth today. There were no forests to anchor sediment, no carpets of grass to soften exposed stone, and no established layer of organic soil. The continent presented itself largely through minerals, fractured rock, and weathered surfaces, with biological activity confined to places where microbes could survive. Soil is more than loose material scattered across the ground. Modern soil is a living system built through repeated interactions among organisms, minerals, water, and decaying organic matter. Microbes break down material, organic residues accumulate, and roots later bind particles together. None of those processes had reached anything resembling their modern scale on Rodinia. Without widespread land plants or complex terrestrial ecosystems, the continent lacked the deep, biologically structured soils familiar today. Rain, therefore, met the ground in a very different way. Drops struck exposed granite, basalt, sandstone, and metamorphic rock, moving across surfaces that had little organic material to slow them. Water entered cracks, reacted with minerals, and carried dissolved elements toward streams and shallow basins. The journey from mountain to ocean could happen with far less biological interference than it does across a forested landscape. Among the materials transported in this runoff were silica, phosphorus, iron, and other elements essential to microbial metabolism. Their movement connected the weathering of continental rock with the chemistry of coastal waters. Every rainfall event could loosen mineral material from exposed surfaces and deliver part of it toward the ocean, where microscopic communities could use whatever nutrients became available. This created an unusual relationship between land and sea. The continent appeared biologically sparse, yet its exposed minerals were supplying ingredients to the places where life was concentrated. A modern forest captures nutrients within leaves, wood, roots, and soil. Rodinia had no comparable biological reservoir across most of its surface. Minerals could therefore remain more directly connected to the cycle between rock, water, and ocean sediment. Weathering also changed the rocks themselves. Carbon dioxide dissolved into rainwater, forming weak carbonic acid that reacted with silicate minerals. These reactions released dissolved ions and altered the composition of water flowing toward the sea. Over immense spans of time, such weathering became part of the planetary balance involving atmospheric gases, rocks, oceans, and sediment. The absence of vegetation also changed the physical character of erosion. Without roots gripping the ground, exposed surfaces could be broken apart by temperature changes, water, and mechanical forces. Sediment moved through rivers and coastal environments, settling into layers that could later become records of the ancient landscape. Here, the apparent emptiness of Rodinia takes on another meaning. The continent had little visible biology, yet its mineral surfaces were participating in the chemistry that sustained the microbial world offshore. Rock became a source of nutrients, rainfall became a carrier, and the ocean received materials released directly from the exposed crust. The dead land, in that sense, was part of a larger living system. Its lack of vegetation allowed geological processes to operate with unusual directness, linking the weathering of continents to the productivity of shallow seas. Across Rodinia, stone and water were already exchanging the ingredients of life, even where the land itself showed almost no sign that life was present. This distinction mattered across exposed continental surfaces. At the edges of Rodinia, Where sunlight entered shallow water, the ancient ocean carried its greatest concentration of biological activity. Nothing there would have looked like a modern reef. The living structures were built from films of microscopic organisms, layered over sediment and shaped by currents, tides, minerals, and light. Cyanobacteria were among the most important inhabitants of these environments. Each was a single cell, lacking a nucleus, yet communities of them could transform an entire shoreline. They captured sunlight through photosynthesis and used that energy to build organic matter. In doing so, they released oxygen into the surrounding water, adding another small contribution to the atmospheric oxygen that had been accumulating for immense spans of geological time. Their communities could also leave physical structures behind. As microbial mats spread across shallow surfaces, sticky substances produced by the organisms trapped grains of sediment. New layers formed above older ones, creating domes, columns, and finely banded structures known as stromatolites. Some of the oldest known stromatolites date to roughly 3.5 billion years ago. Their descendants still build similar formations today, including those found around Shark Bay in Western Australia. A stromatolite was therefore a kind of geological collaboration between living cells and moving sediment. The microbes occupied the upper surface, where sunlight reached them, while mineral grains accumulated around the mat. Layer followed layer as the community grew and sediment was captured. Over long intervals, these structures became part of the rock record, preserving evidence of biological activity from worlds separated from the present by billions of years. The shoreline itself became a record of their patient activity. The work of these organisms extended far beyond the shallow water. Oxygen released during photosynthesis reacted readily with dissolved iron carried through the ocean. Iron that had remained suspended in seawater could be converted into solid minerals and removed from circulation. This changed the availability of iron throughout marine environments and altered the balance of elements moving between water and sediment. As oxygen and iron interacted, another microbial community gained greater influence in deeper waters. Sulfate-reducing bacteria used sulfur compounds during their metabolism and produced hydrogen sulfide. Where oxygen was scarce, this chemistry could become especially important. The resulting sulfide reacted with metals and altered the chemical character of deep marine sediments, contributing to the conditions associated with the Canfield Ocean. The same biological activity that transformed sunlight into chemical energy was therefore changing the ocean around it. Cyanobacteria were releasing oxygen into waters that contained abundant, dissolved materials, while other microbes responded to the new chemical opportunities created by those changes. One community could alter the environment in ways that opened space for another. Across the coastal margins, these interactions repeated across countless generations. Microbial mats grew, trapped sediment, exchanged gases, and modified minerals. Their bodies were microscopic, yet their combined influence reached into the chemistry of oceans and atmosphere. The ancient shoreline carried no forests or animal communities, yet it was filled with biological processes operating at a planetary scale. What appeared to be an empty coastline was therefore a densely occupied microbial landscape. Its inhabitants left layered structures in stone, altered the chemistry of seawater, and helped determine which elements remained available to life. Their world was measured in cells and mineral grains, while its effects extended across oceans and through geological time. There is a strange way to measure a billion years. Imagine watching the continents shift, oceans circulate, and life continue its patient work, then compressing all of those changes into a single geological interval. For much of the 20th century, The middle Proterozoic seemed to offer remarkably little drama by comparison with the chapters around it. Paleontologist Martin Brazier eventually gave this impression a memorable name, the boring billion. The phrase emerged from a growing recognition among geologists that this interval lacked the dramatic biological, geological, and climatic events that defined many other chapters. It was a useful shorthand for apparent stasis, even though the Earth beneath that label continued to operate through countless interacting systems. The phrase referred broadly to the stretch from about 1.8 billion to 800 million years ago. Geological records from this interval often seemed unusually stable. Carbon isotope measurements, which can preserve clues about the global movement of carbon between living organisms, oceans, atmosphere, and rocks, showed a long pattern with relatively little variation. To geologists searching for major shifts, that steadiness looked almost like a pause. Other chemical records added to the impression. Isotopes of sulfur, molybdenum, chromium, and strontium also suggested a world whose surface chemistry changed within a comparatively narrow range. The evidence pointed toward prolonged limitations in nutrients, oxygen, and other ingredients that can influence biological expansion. Even the climate appeared restrained compared with the dramatic glaciations recorded elsewhere in Earth's history. The continents were still moving, however, and the crust continued to breathe through cycles of uplift, burial, melting, and erosion. While the supercontinent Columbia had assembled earlier and later fragmented, and Rodinia was being constructed through collisions among continental blocks. Mountains rose and eroded, oceans opened and narrowed, and the crust continued to exchange material with the mantle. The pace and character of these processes may have differed from modern patterns, yet Earth remained geologically active. A billion years is also difficult for the human mind to hold. The first recognizable animals appeared only hundreds of millions of years ago. From their emergence to the present is therefore shorter than the interval carrying the boring billion. Even the entire Phanerozoic eon, the chapter of Earth history dominated by abundant visible life, occupies less time than this prolonged middle stretch. For a long time, the apparent lack of major change encouraged a convenient story. Evolution seemed stalled, climate seemed settled, and the chemistry of the surface appeared locked into place. The planet could be imagined as spending an age in the same position, turning beneath the same subdued sun, while microbial life persisted around the margins of its oceans. That picture has gradually become harder to defend. Molecular fossils, unusual chemical signatures, microscopic structures, and improved geological dating have revealed activity that older records could not easily display. Complex cells were diversifying during parts of this interval, while changes in metabolism and nutrient use unfolded at scales too small to leave spectacular landscapes or obvious fossil monuments. The boring billion, therefore, became a lesson in the limits of visible evidence. A planet can appear unchanged when the important transformations are occurring inside cells, sediments, minerals, and chemical cycles. The surface may offer few grand events, while countless molecular experiments continue underneath the threshold of ordinary observation. The oxygen story had already begun, yet its progress seemed to settle into a narrow corridor. After the great oxygenation event, oxygen entered the atmosphere in amounts that permanently changed Earth. During the opening of the boring billion, however, atmospheric oxygen remained only a small fraction of modern levels, perhaps around 1 to 2% of today's abundance. Even after another billion years, estimates suggest it had reached only a few percent of modern levels. This prolonged plateau forms the heart of the oxygen paradox. Photosynthetic cyanobacteria were still producing oxygen day after day across shallow waters and microbial communities. Their activity should have allowed oxygen to accumulate. Something else was continually removing the ingredients needed for that production to expand. One important part of the answer rested in the chemistry of the deep ocean. Sulfidic waters could bind important trace metals into minerals, drawing them away from regions where surface microbes depended on them. Molybdenum was especially important. Certain bacteria require molybdenum to carry out nitrogen fixation, a process that converts atmospheric nitrogen into forms usable for biological growth. Without sufficient access to this metal, cyanobacteria faced a limit on how much new biomass they could produce. Phosphorus presented another constraint. It is a fundamental nutrient for living cells, involved in energy transfer, genetic material, and cell membranes. When phosphorus becomes scarce, microbial productivity can weaken even when sunlight and carbon remain available. In a world where nutrients were being shuffled into deep sediments and chemically restricted waters, surface communities could reach a ceiling imposed by the ocean itself. The feedback became self reinforcing. Reduced microbial growth meant less oxygen entered the atmosphere. Lower oxygen allowed extensive oxygen poor and sulfide rich waters to persist. Those conditions encouraged chemical processes that continued removing nutrients and trace metals from circulation. With fewer essential resources returning to sunlit waters, the organisms responsible for much of Earth's oxygen production remained constrained. This was a planetary system operating within its own limits. Sunlight reached the ocean's surface, carbon dioxide was available, and photosynthesis could proceed, yet the supply of critical nutrients could not expand fast enough to transform the atmosphere. Each part of the cycle supported another part, holding the whole system within a narrow chemical range. The consequence reached far beyond the chemistry of air. Low oxygen limited the energy available to organisms with demanding metabolisms, while nutrient scarcity restricted the expansion of the microbial communities that might otherwise have altered the planet more rapidly. Complex life required more than the presence of oxygen itself. It depended on an entire network of environmental conditions developing together. For hundreds of millions of years, that network remained incomplete. Oxygen was present, sunlight was abundant, and life had already discovered photosynthesis, yet the necessary pieces remained separated by the chemistry of the ocean. Earth carried many of the ingredients associated with later biological complexity, but they were distributed across a system that prevented them from reinforcing one another. The result was an unusual kind of stability. Life continued to alter its surroundings, while those altered surroundings constrained life in return. The atmosphere and ocean became partners in maintaining a long-lived equilibrium, allowing oxygen to persist without allowing it to become abundant. For nearly a billion years, the planet remained caught within that delicate exchange, with possibility held inside chemistry. A thin layer of sunlight reached the upper ocean, carrying energy into waters where the chemistry of life was already constrained. The surface had access to light and carbon, yet several elements essential for growth were becoming difficult to obtain. The problem was less a lack of ingredients on Earth than their confinement in the wrong places. Phosphorus was among the most important. Every living cell depends on it for energy transfer, cell membranes, and the construction of genetic material. In a more oxygenated ocean, phosphorus can move through a cycle linking deep water, sediments, and sunlit surface waters. During the Canfield Ocean, much of that circulation was disrupted. Phosphorus became associated with sediments and was less available where photosynthetic microbes needed it most. Iron faced a different fate. Photosynthetic organisms require iron in several enzymes involved in capturing energy and processing nutrients. In deep water containing abundant sulfide, dissolved iron could react with sulfur and become locked into iron sulfide minerals. That process removed iron from circulation before it could return to the upper ocean through mixing. Molybdenum was another scarce piece of the puzzle. Certain bacteria depend on this trace metal for nitrogen fixation, the process that converts atmospheric nitrogen into a form biological cells can use. With limited molybdenum available, access to usable nitrogen became more difficult. A cell could have sunlight and carbon around it, yet still lack the materials needed to expand. These shortages mattered because microbial life had to build itself from a limited chemical budget. When phosphorus was scarce, growth slowed. When iron was restricted, important metabolic reactions became harder to sustain. When molybdenum was limited, nitrogen acquisition became constrained. Each shortage touched another part of cellular chemistry, creating a surface ocean where productivity could persist without easily accelerating. The effects reached into the architecture of life itself. Complex multicellular organisms require large amounts of energy to maintain many cells, coordinate their activities, repair damage, and move materials through tissues. Such demands depend on reliable supplies of nutrients and oxygen. A nutrient poor surface ocean could sustain organisms with modest requirements, while leaving little ecological room for forms of life that needed a much larger energetic budget. Sediments became part of this long restraint. Material carried from land settled through coastal waters and accumulated on the seafloor. Some of it remained buried for immense spans of time, removing phosphorus and other elements from immediate circulation. Chemical reactions within the sediment could further alter what returned to the water above. The ocean, therefore, became a place where nutrients were continually transformed, stored, and separated from the communities that needed them. This was a different kind of environmental limit from darkness, cold, or physical isolation. The surface received sunlight every day, and microbial life continued its intricate exchanges. What constrained it was the availability of particular atoms and forms that cells could actually use. For an organism living in this world, survival could depend on making extraordinary use of a very small chemical allowance. The surface communities endured through efficiency, recycling, and adaptation to scarcity, passing scarce elements from one generation of cells to another with unusual economy. Their existence kept biological activity moving through the oceans, while the deeper chemistry prevented that activity from expanding into the larger energetic systems required for complex, multicellular life. Somewhere in the shallow waters of the boring billion, two forms of life came together in a way that changed the future of the planet. The event left little spectacle in the rocks, yet its consequences reached into every complex organism that would ever appear. A partnership began at a scale far too small for any ancient landscape to record. One ancient archaeon encountered a bacterium and somehow brought it inside its own cell. The bacterium was not destroyed. It remained alive within its host, and the two organisms began exchanging resources. Over generations, the relationship became permanent. The former bacterium gradually lost much of its independence, while its host gained an internal source of energy. Its descendants became mitochondria, the structures that power the cells of animals, plants, fungi, and many other eukaryotic organisms. This process is known as endosymbiosis. It was a merger between distinct living systems, creating something larger than either partner could have achieved alone. The internal bacterium could process nutrients and generate energy efficiently, while the host provided protection and access to resources. Their partnership transformed the economy of the cell. That transformation mattered because energy sets limits on what a cell can build. A cell with greater access to usable energy can maintain more internal machinery, support larger volumes, and manage greater quantities of genetic information. It can also divide labor among specialized structures inside its boundary. The appearance of these capabilities opened a new biological scale. Evidence for eukaryotic cells appears in rocks roughly 1.5 to 1.7 billion years old, including fossils and chemical traces from parts of Australia. These records are modest compared with the spectacular fossils of later eras, yet they reveal a profound change in cellular organization. Life had acquired a new architecture. The difference was deeper than size. Eukaryotic cells contain a nucleus that separates genetic material from much of the surrounding cellular machinery. They also contain internal compartments that allow different chemical tasks to occur in different places. This organization created greater control over complex cellular processes and provided room for biological systems to become increasingly elaborate. Another consequence emerged through reproduction. Once cells possessed this greater internal complexity, lineages could develop mechanisms for combining genetic material from different individuals. Sexual reproduction did not appear as a single completed invention, and its early history remains difficult to reconstruct, but the cellular foundations for genetic mixing became possible within this broader transition. Biologist Ernst Mayr described the origin of the eukaryotic cell as perhaps the most important and dramatic event in life's history. The description captures the scale of what happened, even though the event itself was microscopic. There was no mountain range to mark it, no ocean basin to preserve its arrival, and no large organism to leave an unmistakable impression. The transformation unfolded inside cells while the larger planet appeared comparatively stable. Across immense spans of time, these new cellular partnerships multiplied, changed, and diversified. The world still belonged to microbes, yet one kind of microbe had acquired a new internal arrangement. Within that arrangement rested the capacity for larger bodies, specialized tissues, complex reproduction, and eventually every forest, animal, and human life that would follow. The future of complex life began as a cellular relationship, preserved through generations until the partners could no longer be separated. The mitochondrion gave the eukaryotic cell a new kind of internal economy. Descended from a bacterium that became a permanent partner, it could generate energy through aerobic respiration with far greater efficiency than fermentation. Estimates vary with conditions, but the difference was substantial enough to alter what a cell could afford to build and maintain. Energy that once had to be stretched across basic survival could now support larger volumes, more internal structures, and more demanding chemical work. That advantage changed the scale of cellular possibility. Early eukaryotes remained single-celled organisms, yet their cells possessed features that opened unfamiliar ways of living. Some could move actively through water. Others could engulf neighboring cells and digest them internally. Certain lineages could capture light, while others relied on respiration for energy. Their genetic material was enclosed within a nucleus, separated from much of the surrounding cellular machinery by a membrane. The nucleus added another layer of control. Genetic material could be organized and protected, while molecular machinery regulated when particular genes were active. This allowed cells to coordinate many processes at once. Much later, those principles would become essential to multicellular bodies, where related cells could carry the same genome while using different portions of it. A muscle cell, a nerve cell, and a blood cell can therefore develop different identities while retaining the same basic genetic library. The story gained another remarkable turn when one eukaryotic cell incorporated a cyanobacterium. Again, the captured organism remained alive and became part of its host. Its descendants evolved into chloroplasts, the light capturing organelles found in plants and many algae today. The arrangement gave those cells a second internal power system, one able to convert sunlight into chemical energy. The partnership eventually helped establish the cellular foundation of forests, grasses, flowering plants, and countless aquatic organisms. These mergers changed biology through cooperation between cells that once lived independently. The descendants of those partnerships still carry clues to their origins. Mitochondria retain small genomes of their own, while chloroplasts also preserve genetic material separate from the nucleus. Their internal machinery resembles that of bacteria in several important ways. Even their methods of reproduction retain echoes of their former independence. For a long interval, however, the eukaryotic world remained microscopic. Complex cells had appeared, but the planet still lacked the towering forests and moving animals that would later become familiar. The new cellular architecture needed immense spans of time to spread through different lineages and acquire further innovations. Its significance was already present in the organization of each cell, waiting in the chemistry of membranes, genes, proteins, and energy transfer. Every insect, tree, whale, and human ultimately carries this history within its cells. The same basic partnership between an ancestral host and an engulfed bacterium persists inside modern life. Another ancient merger supplies the machinery for photosynthesis in plants. What looks today like a seamless unit is therefore a record of cooperation preserved at microscopic scale for billions of years. The boring billion gave life something more enduring than a dramatic fossil. It provided a new cellular design, one capable of managing greater energy, greater information, and greater internal complexity. The architecture was small enough to escape the notice of ancient landscapes, yet large enough in consequence to reshape the future of the living Earth. A continent can change an ocean without touching its water directly. Around 1.46 billion years ago, the supercontinent Nuna, also known as Columbia, began to fragment, sending its continental pieces outward and reshaping the margins where land met sea. The movement unfolded across hundreds of millions of years, yet its consequences reached into the chemistry of shallow water, where sunlight, minerals, and oxygen could meet in unusually favorable combinations. As the supercontinent dispersed, the length of shallow continental margins expanded dramatically. Geological modeling, published in 2025, found that passive margins more than doubled during the breakup, eventually reaching about 130,000 kilometers, which is about 81,000 miles. These shelves were far more than narrow borders around continents. They created broad marine platforms where sunlight could penetrate, sediments could accumulate, and nutrients from weathered land could enter productive waters. Their importance came from their position between two worlds. Deep ocean water could remain deprived of oxygen and rich in reduced chemicals, while shallow seas received sunlight and fresh mineral material from nearby continents. More shelf area meant more places where these conditions could persist side by side. The expanded margins, therefore, offered long-lived habitats where oxygen could accumulate locally and where complex cells had greater access to the resources needed for growth. Sediment also gathered across these shelves, preserving chemical exchanges between rock, seawater, and microbial communities. Each margin became a broad interface where geological change could be translated into biological opportunity over time. The tectonic rearrangement also altered the planet's carbon system. As Nuna broke apart, the global pattern of plate boundaries changed. The study's models indicate that volcanic carbon dioxide release from the solid Earth declined substantially as subduction zones shortened. At the same time, more carbon was stored within oceanic crust. Seawater circulated through fractures in the crust, interacted with hot rocks, and helped convert dissolved carbon into carbonate minerals. Carbon that might otherwise have returned to the atmosphere became part of the geological structure of the ocean floor. That shift carried climatic consequences. Lower volcanic carbon dioxide release, combined with greater geological carbon storage, favored a cooler and more stable climate. Cooler conditions could reduce the intensity of chemical reactions that consume atmospheric oxygen, allowing oxygen to accumulate more readily. The effect was gradual, measured across immense spans of geological time, yet it connected processes deep inside Earth with changes occurring at the ocean surface. The expanding margins also created a larger stage for biological experimentation. Early eukaryotic organisms had already appeared, but their world remained constrained by limited oxygen and nutrients. New shallow seas offered repeated environments where these cells could encounter minerals, sunlight, and other organisms under more favorable conditions. Instead of one isolated habitat, there were many coastal settings distributed around fragments of a changing continent. Nuna's breakup therefore becomes part of the story through geography as much as geology. The continents moved apart, and the ocean gained new edges. Those edges increased the space available for productive shallow seas, while tectonic carbon cycling altered the atmosphere and climate above them. The long interval, called the boring billion, contained a planet in motion, with plate boundaries, ocean chemistry, and living cells gradually adjusting to one another. Rodinia did not vanish all at once. Its great continental mass began to split into separate pieces between about 750 and 700 million years ago, as heat rising from deep within Earth weakened the crust above it. One proposed driver was a mantle plume beneath what is now Canada, where unusually hot material rose toward the surface and helped initiate a chain of fractures. The continent began opening along long zones of broken rock, allowing new ocean floor to form between the separating blocks. Across those fractures, magma rose into the widening gaps, leaving a growing record of tectonic separation in the rocks themselves. Each new rift changed the geography of the planet, while the chemistry of the atmosphere responded in ways that were harder to see. Mid-ocean ridges rose along the newly forming seafloor, where molten rock reached the surface and created fresh crust. These ridges normally release carbon dioxide through volcanic activity, yet geological reconstructions indicate that carbon dioxide emissions were unusually low during this interval. At the same time, the breakup exposed enormous areas of fresh volcanic rock to rain and weathering. Fresh silicate minerals have a particular relationship with the atmosphere. Rainwater absorbs carbon dioxide and becomes mildly acidic, allowing it to react with exposed rock. During this process, carbon is carried into rivers and eventually reaches the ocean, where some become stored in carbonate minerals. On a continent covered by old, weathered surfaces, this process proceeds at a different pace. Newly exposed volcanic provinces can provide abundant reactive minerals, creating a stronger draw on atmospheric carbon dioxide. Rodinia's fragmentation therefore established two carbon sinks at once. Volcanic release from the ocean ridges was reduced, while chemical weathering on land accelerated. Neither process needed to transform the climate overnight. Their influence accumulated across immense spans of time, gradually lowering the concentration of carbon dioxide in the atmosphere. Because carbon dioxide helps regulate how much heat Earth retains, a sustained decline could shift the balance toward cooler global conditions. The first consequences appeared at the highest latitudes, where ice had the best chance to survive from one season into the next. Once snow and ice covered more ground, another physical process entered the story. Ice is highly reflective. Instead of absorbing much of the incoming sunlight, bright frozen surfaces send a greater share of that energy back toward space. Scientists call this the ice albedo feedback. That feedback could amplify the original cooling. Lower temperatures allowed ice to persist across larger regions. More ice increased planetary reflectivity. Greater reflectivity reduced the solar energy absorbed by Earth, which encouraged still more cooling. A modest shift in atmospheric chemistry could therefore become connected to a much larger transformation of the climate system. The continents were still moving, oceans were still forming, and the planet's interior continued to exchange heat with the surface. Yet the balance among these processes had changed. Rodinia's breakup had opened fresh rock to weathering while altering volcanic carbon release, creating conditions in which cooling could reinforce itself. For the first time in this long geological story, the frozen state of the planet could become self-sustaining. The same world that had endured immense chemical stability was entering a climate regime shaped by feedback, reflection, and the gradual removal of atmospheric carbon dioxide. A change at the poles can remain confined to high latitudes for a long time until a threshold is crossed and the climate begins feeding the change back upon itself. Around 717 million years ago, that threshold appears to have been reached during the Sturtian glaciation. Ice expanded outward from the polar regions, reflecting sunlight and reducing the heat absorbed by the surface. The colder ocean then became easier to freeze, allowing the bright surface to spread farther. A planetary transformation was taking shape through a chain of physical responses. Geological evidence places this frozen world far beyond the regions where glaciers normally survive. Among the most revealing clues are dropstones, rocks transported by floating ice and released into marine sediments when the ice melted. Some of these deposits formed at latitudes close to the equator. Their presence means that ice reached places where modern climate would make permanent glaciation almost unimaginable. Another line of evidence comes from paleomagnetism. As certain sediments and volcanic materials formed, magnetic minerals within them aligned with Earth's magnetic field and preserved its ancient direction. Scientists can use those orientations to estimate the latitude where the rocks originally formed. When glacial deposits from the Sturtian interval show signatures consistent with formation near the equator, the evidence points toward a planet whose frozen regions extended across nearly the entire globe. The temperature change would have been extraordinary. Estimates for average global conditions during the deepest part of the episode reach about 51 degrees below 0 Celsius, or 60 degrees below 0 Fahrenheit. That is colder than modern Antarctica and far below the temperatures normally associated with Earth's ocean surface. At such temperatures, open water would have become increasingly rare, especially as ice reflected more incoming sunlight back toward space. The frozen cover was also substantial. Near the poles, ice sheets may have reached around 1,600 meters, which is about 5,250 feet in thickness. Even around the equator, where sunlight was strongest, estimates suggest hundreds of feet of ice. A planet covered by such a shell would have had an ocean largely separated from the atmosphere, with sunlight reaching only limited regions through the frozen surface. Yet, this was not a brief winter. The Sturtian glaciation persisted for roughly 57 million years, a span long enough for continents to continue drifting beneath the frozen climate and for geological processes to reshape the planet underneath the ice. During that immense interval, volcanic activity could continue because Earth's interior remained hot even while the surface was deeply frozen. Volcanoes released gases into an atmosphere increasingly isolated from the ordinary exchange between air, ocean, and living systems. The resulting world would have been radically different from the warmer planet that preceded it. Ice connected distant latitudes into a single climatic system, while the oceans beneath it became difficult places for sunlight driven ecosystems to persist. Still, Earth retained an active interior, moving continents, circulating rock, and volcanoes that continued to add material to the surface. The frozen planet was therefore alive with geological activity, even while its exposed surface had become almost entirely ice. Under that frozen boundary, chemical reactions continued, minerals moved through cracks, and the deep ocean remained part of a planetary system that had never truly stopped changing. Ice covered the ocean, yet life found ways to remain connected to energy beneath the frozen surface. With sunlight largely blocked, the familiar partnership between light and photosynthesis was severely weakened. Cyanobacteria and early algae could no longer sustain the broad food webs that had depended on surface production. Much of the ocean became a place where biological activity had to rely on sources of energy other than sunlight. One refuge existed far below the frozen surface, around hydrothermal vents on the seafloor. These vents form where heated water rises through fractured crust, carrying dissolved minerals from Earth's interior. When that fluid meets colder seawater, chemical reactions release usable energy. Microorganisms can capture part of that energy and turn it into organic matter, creating communities that do not depend directly on sunlight. Similar ecosystems exist in Earth's deep ocean today, where life gathers around mineral rich fluids emerging from the crust. Other refuges may have occupied the narrow boundary between ice and water. Geochemical models suggest that volcanic activity could have maintained localized meltwater beneath the ice, while geothermal heat may have preserved pockets of liquid water in places where the frozen shell was thinner. These environments would have been small compared with the global ocean, yet their isolation could have given microbial communities unusual evolutionary histories. Populations separated for immense periods would have accumulated different mutations, adapted to different chemical conditions, and gradually become more distinct from one another. For eukaryotic life, that separation may have mattered. The earliest eukaryotes were already carrying a different cellular architecture, including internal compartments and mitochondria that allowed greater control over energy use. During a world divided by ice, isolated populations could drift along separate evolutionary paths. Some may have remained adapted to scarce nutrients and low energy conditions, while others occupied chemically richer refuges near volcanic systems. Genetic differences could accumulate without being constantly mixed through contact between distant populations. The frozen climate also changed the geography of survival. Ice sheets and glaciers created physical barriers between regions, while the underlying ocean retained channels where heat and dissolved chemicals could circulate. Volcanic islands and submarine ridges provided localized sources of warmth and minerals. Each refuge became a small experiment in persistence, with its own chemistry, energy supply, and microbial inhabitants. The duration of the Sturtian glaciation made these processes especially significant. Roughly 57 million years gave isolated populations an immense span for adaptation and divergence. Across that interval, countless generations lived within environments that were separated from one another by frozen ocean. Some lineages would disappear, while others continued through changing conditions, carrying their cellular innovations forward. The surface of Earth during this era offered little opportunity for ordinary photosynthetic ecosystems, Yet the planet's interior continued supplying energy through volcanism and geothermal activity. Life, therefore, survived through a patchwork of refuges, each sustained by a different balance of heat, minerals, water, and chemistry. The frozen world had narrowed the places where organisms could live, while also separating populations for a period almost impossible to imagine. Within those small surviving communities, biological history continued to accumulate, one generation after another. Volcanic ash and mineral dust may also have darkened parts of the ice, increasing local melting and creating small ponds. Within sea ice, concentrated brines could preserve liquid pathways rich in salts and dissolved compounds, offering microorganisms another place to endure. For millions of years, the frozen surface could conceal a world that was still producing heat. Deep inside Earth, radioactive decay and leftover planetary heat continued driving convection through the mantle. Magma rose along mid-ocean ridges, and volcanic systems kept releasing carbon dioxide. The gas dissolved into seawater where it could, then gradually accumulated in the atmosphere through fractures, cracks, and regions where ice allowed exchange between ocean and sky. Ordinary Earth had a way of removing that carbon dioxide. Photosynthetic organisms could incorporate it into living matter, while chemical weathering of exposed rock transferred carbon from the atmosphere into minerals and eventually sediments. Snowball conditions disrupted both pathways. With sunlight largely blocked, photosynthesis became extremely limited. Glaciers covered much of the land, reducing the contact between rainwater and freshly exposed silicate rock. The planet had entered a state in which its principal greenhouse gas could accumulate for an extraordinary span of time. Each volcanic pulse added another small amount. No single eruption needed to transform the climate. The change emerged from accumulation, as carbon dioxide remained in circulation while removal processes operated at only a fraction of their usual strength. The atmosphere became increasingly capable of trapping outgoing heat. Eventually, the greenhouse effect grew strong enough to challenge the reflective ice surface that had helped preserve the frozen state. The first important melting could occur near the equator, where volcanic warmth and accumulated greenhouse gases made the ice especially vulnerable. Once darker ocean water appeared, it absorbed more sunlight than reflective ice. That additional energy encouraged further melting, exposing still more dark water. The relationship became self-reinforcing. Less ice meant more absorbed sunlight, and more absorbed sunlight meant less ice. The transition from frozen planet to warm planet could therefore unfold with extraordinary speed on a geological scale. Climate models suggest that temperatures may have risen by roughly 122 degrees Fahrenheit during the deglaciation, although the exact magnitude remains uncertain. The planet could move from conditions near 60 degrees Fahrenheit below zero to temperatures approaching 120 degrees Fahrenheit in some reconstructions. Such numbers describe a climate transformed from one extreme to another across a span that was brief compared with the millions of years of freezing that came before. As the ice retreated, evaporation surged. Water vapor entered the atmosphere in enormous quantities, while carbon dioxide made rainfall increasingly acidic. Rain began reaching freshly exposed rock across continents that had been shielded beneath ice. Silicate minerals reacted with carbonic acid, drawing carbon dioxide from the atmosphere and carrying dissolved products toward the ocean. The newly exposed land became a vast chemical surface. Rivers transported minerals and nutrients into warming seas, while weathering accelerated under intense rainfall. The same carbon dioxide that had helped end the global freeze was now being consumed by the newly awakened geological cycle. Earth had passed through an extraordinary climatic reversal, with ice giving way to water, rainfall, and a planet once again open to the atmosphere. Along newly opened coastlines, ice that had reflected sunlight disappeared into expanding seas, while clouds gathered above water warmed after ages of confinement. Storm systems could become powerful, feeding on vapor. Rainfall reached regions that had experienced little surface weathering for millions of years, turning exposed rock into reservoirs for minerals and nutrients. The climate was becoming chemically active again, with atmosphere, ocean, and continents exchanging matter. The first thaw did not settle Earth into a lasting climate. Around 660 million years ago, the Sturdian glaciation had ended, leaving oceans and continents exposed once again. Yet the balance between atmosphere, rock, ocean, and ice remained fragile. Carbon dioxide began moving through the planetary system under new conditions, while weathering and volcanic activity continued reshaping the climate. The Great Freeze had passed, though the world had not finished moving through its extremes. Around 650 million years ago, another descent began. The Minoan glaciation spread across the planet, locking the oceans beneath ice for roughly 15 million years. This second snowball Earth was shorter than the Sturtian event, yet geological evidence indicates that its reach was again global. Ice advanced across continents, sea ice extended toward the tropics, and much of the surface became enclosed within a frozen climate system. One of the clearest clues appears in a geological pairing found across widely separated continents. Glacial deposits are directly overlain by layers called cap carbonates, thick accumulations of limestone that formed rapidly when the oceans became warm and rich in carbon dioxide. Their position is especially revealing. A frozen landscape lies below, followed abruptly by rocks produced under warm, chemically active seas. The boundary preserves a record of a planetary climate changing from one extreme toward another. Each cycle also altered the ocean itself. During prolonged freezing, circulation was greatly reduced, allowing dissolved materials to accumulate in particular environments. Iron could build up in seawater and sediments, while phosphorus became associated with minerals and organic matter. Sulfide chemistry changed as the availability of oxygen and other chemical reactants shifted. When melting returned, fresh water and newly exposed minerals entered the oceans, disturbing these old chemical arrangements. Phosphorus was especially important because it could become available to organisms after being released from sediments. Iron also became mobile in changing waters, allowing it to participate in reactions that had been limited during prolonged anoxia. Sulfide concentrations could decline as oxygen began penetrating environments that had previously remained chemically isolated. The ocean was therefore becoming a different chemical landscape, with materials moving between sediments, seawater, and the atmosphere in new combinations. The repeated passage through freezing and thawing acted like a planetary reset. Established communities were reduced, habitats disappeared, and chemical resources that had accumulated for immense spans of time were redistributed. Every thaw opened fresh surfaces and altered nutrient pathways. Every renewed freeze narrowed the available refuges and divided populations into isolated pockets. Evolution continued inside these changing boundaries, shaped by which organisms could endure scarcity, instability, and separation. Survival across both global glaciations demanded unusual flexibility. Some organisms could tolerate long periods with little energy, while others occupied specialized refuges where liquid water and chemical energy remained available. Populations separated for millions of years could accumulate genetic differences, developing distinct adaptations while the surrounding planet repeatedly shifted between extremes. The survivors carried more than continuity. They carried biological solutions refined through repeated environmental upheaval, preserved within cells that had endured a world repeatedly transformed by ice, water, chemistry, and time. For more than 1 billion years, oxygen had moved through Earth's systems in limited pulses, appearing in local environments before being consumed by minerals, sulfur compounds, and other chemical sinks. After the great glaciations, that balance began to shift. Around 635 million years ago, oxygen levels rose as the oceans and atmosphere entered a new chemical regime. Some researchers describe this broader transformation as part of the Neoproterozoic oxygenation event, a prolonged transition that changed the availability of oxygen across marine environments. The increase was gradual on a human scale, yet profound across geological time. Atmospheric oxygen remained below its modern abundance for much of this transition, but the direction of change mattered enormously. More oxygen reached seawater, while newly exposed continents supplied minerals to rivers and coastal basins. The boundary between oxygen-rich surface waters and chemically reduced deep waters became increasingly unstable. Sulfide, once abundant in many deep marine settings, began reacting with oxygen. Those reactions altered the chemistry of the water and changed where sulfur could remain dissolved. Iron underwent a similar transformation. When oxygen penetrated deeper into the ocean, dissolved iron could be converted into oxidized forms that settled into sediments. Phosphorus, meanwhile, could be released from previously stable mineral associations under changing chemical conditions, allowing more of this essential nutrient to circulate through marine ecosystems. The old structure of the ocean began losing its grip. Deep water could exchange more freely with surface water, carrying dissolved substances between regions that had once behaved almost like separate chemical worlds. Oxygen did not need to fill every part of the ocean at once. Even partial expansion into deeper environments could alter nutrient cycles, microbial communities, and the availability of energy for organisms capable of using aerobic respiration. For eukaryotic cells, this changing chemistry mattered deeply. Their mitochondria could extract substantial energy from oxygen, supporting cellular activities that required larger and steadier energy budgets. Nutrients, such as phosphorus, iron, and molybdenum, became increasingly available in environments where oxygen and circulation could keep them moving through biological and geological cycles. The ocean was becoming a more connected chemical system, with ingredients that had once been locked away entering pathways that life could use. Molybdenum carried a particular importance because several enzymes involved in nitrogen fixation depend on it. Greater availability of this metal could help expand biological access to nitrogen, an element required for proteins and genetic material. Phosphorus supported energy transfer and cellular membranes, while iron remained central to many enzymes involved in respiration and photosynthesis. Together, these elements formed part of the chemical foundation required by increasingly complex cells. The transformation unfolded across immense spans of time, yet its consequences reached into every later chapter of life. The ocean no longer held the same rigid separation between nutrient, poor surface waters and chemically isolated depths. Oxygen, minerals, and nutrients could circulate through a wider range of habitats. A planetary system that had constrained complexity for ages was becoming more permissive. Nothing about the change required a single instant. It emerged through countless chemical reactions, shifts in circulation, weathering, burial, and release. Still, viewed across geological time, the transition resembles the opening of a door that had been closed for an extraordinary duration. The ingredients for complex life were becoming available together within an ocean whose chemistry could finally support their use. The Ediacaran seafloor held forms that seem almost improvised, as though life were testing unfamiliar ways of becoming large. Between roughly 635 and 541 million years ago, the fossil record begins preserving a community, unlike the animals that dominate later chapters of Earth's history. These organisms are known as the Ediacaran fauna, named after the Ediacara Hills of South Australia, where several important fossils were discovered. Their bodies were often flat, broad, and divided into repeating sections. Some resembled fronds, while others looked like quilted cushions spread across the sediment. Many had no obvious head, mouth, eyes, or limbs. Their surfaces may have absorbed dissolved nutrients or hosted microbial communities that supplied food. The ocean floor itself became part of their living space, and their bodies interacted with it across broad areas rather than moving through it like later animals. Dickinsonia offers one of the clearest glimpses into this unfamiliar biology. Some specimens reached about 1.4 meters, which is about 4.6 feet, making this organism enormous compared with many of the small creatures living around it. Its oval body carried repeated segments arranged along a central axis, giving it the appearance of a soft, patterned mat. Evidence of movement across ancient microbial surfaces suggests that Dickinsonia could shift position, leaving traces as it traveled over the seafloor. Chemical evidence from some fossils also indicates that at least some individuals consumed organic matter, giving this strange body a place within the developing food web. Charnia followed a different design. Its body rose from the seafloor in a branching frond, resembling a leaf more than a familiar animal. Anchored at its base, it extended into the water column, where currents could bring dissolved material and suspended particles past its surface. Its construction shows that multicellular life had begun experimenting with height, shape, and surface area, creating bodies suited to extracting resources from moving seawater. The Ediacaran organisms also reveal how little the first large bodies needed to resemble modern animals. Their tissues were soft, leaving few durable structures behind. Without shells, bones, teeth, or other mineralized parts, their presence survives mainly through impressions preserved in sediment. Some fossils record entire communities across ancient surfaces, allowing paleontologists to reconstruct a seafloor occupied by organisms with forms that have no close modern equivalent. Their presence also changed the physical texture of the seafloor. Broad bodies pressed into microbial films, creating patches where sediment was disturbed and nutrients could be redistributed. In places, overlapping impressions suggest dense communities rather than isolated individuals. The fossils preserve a moment when animal life had become visible across the ocean floor, even though its members still belong to an experimental stage of biological design. For roughly 100 million years, these communities persisted through changing conditions. Their long existence shows that evolution had already produced a stable experiment in multicellular life before the later diversification of animals. They occupied space, gathered resources, reproduced, and interacted with their surroundings, even though their bodies followed designs that would largely disappear. There were no obvious jaws moving through these communities, no armored shells crossing the sediment, and no familiar limbs probing the water. Life had reached a new scale, yet its architecture remained wonderfully open-ended. Across the newly oxygenated seas, soft bodies spread over microbial surfaces, turning the seafloor into a living landscape whose inhabitants belong to a chapter of evolution unlike almost anything alive today. Around 541 million years ago, the fossil record begins to change with unusual speed. Across sediments laid down in ancient seas, bodies appear with recognizable divisions, protective coverings, grasping limbs, feeding structures, and sensory organs. The world of animals had acquired a new vocabulary. What had once been represented mostly by soft impressions now included forms built for movement, feeding, defense, and interaction. Trilobites moved across the seafloor on jointed limbs, their armored bodies divided into repeating sections. Elsewhere, anomalocaridids carried flexible appendages toward their mouths, becoming some of the largest predators of their communities. Brachiopods anchored themselves to the sea floor and filtered nourishment from passing water. Among these creatures were early chordates, slender animals whose internal structures belonged to the lineage that would eventually include every fish, amphibian, reptile, bird, and mammal. The appearance of these forms can seem abrupt when preserved in stone, although their origins reach much farther back. Complex cells had already spent immense stretches of time refining internal compartments, genetic control, and energy management. The oceans had also been reshaped by the changes that followed the great glaciations. Nutrients became more available, oxygen could support greater metabolic demands, and ecological relationships grew increasingly intricate. Evolution had accumulated possibilities long before fossils began displaying them so clearly. Eyes added another dimension to this new world. Once light could be detected with greater precision, animals could respond to movement and distance across the seafloor. Predation then became more than a matter of chance encounters. Speed, armor, burrowing, concealment, and sharper senses could each become useful traits. Every advantage created pressure for another response, and those responses created new opportunities in return. Shells and mineralized structures brought another change. A body protected by a hard covering could occupy exposed surfaces while resisting damage from predators and shifting sediment. Limbs allowed animals to crawl, grasp, and manipulate their surroundings. Mouths made active feeding possible, while specialized body regions began separating different tasks. Animal anatomy was becoming modular, with one part suited for movement and another for feeding or sensing. The Cambrian explosion, therefore, represents a transformation in the visible architecture of life, compressed into roughly 20 million years. 20 million years remains an immense span for a human lifetime, yet it is brief beside the billion years of evolutionary preparation that came before it. In the geological record, that contrast makes the diversification appear almost instantaneous. The long interval, sometimes called the boring billion, now takes on a different meaning. Its oceans were chemically constrained, its climates repeatedly disrupted, and its early complex cells were developing within limits that shaped what could follow. Tectonic changes altered the chemistry of seas and continents. Global freezes tested survival. Each episode changed the resources, habitats, and pressures available to living organisms. The animals that emerged from this history carried traces of that long preparation in every cell and body plan. Their appearance was a threshold reached after countless smaller changes had accumulated. The Cambrian seas became a place where evolution could build with new materials, new energy budgets, and new ecological possibilities. And the first great animal communities began filling those spaces with movement, form, and variety. Fossils from this interval preserve the transition with unusual clarity, allowing later generations to see how quickly biological diversity can expand when environmental opportunity, inherited complexity, and ecological interaction begin reinforcing one another across an entire ocean. Somewhere between the rocks that remain and the rocks that vanished lies one of the greatest gaps in Earth's history. Nearly a billion years of geological evidence has been scattered, altered, buried, or erased, leaving scientists to reconstruct an era from fragments rather than a continuous record. The missing pages are not empty of meaning. They are clues to what happened before the animal world became abundant. Even a single surviving grain of mineral can carry a chemical signature inherited from conditions that no longer exist. A thin layer of stone can therefore preserve information far older than the landscape surrounding it today, even now. The Grand Canyon offers one of the clearest views of this problem. Within its layered walls, the 60-mile formation preserves ancient sedimentary rocks beneath much younger Cambrian deposits. Between them lies the Great Unconformity, a boundary where immense stretches of geological time are absent. In some places, roughly 250 million years of history disappeared from the exposed sequence. The rocks above remain, the rocks below remain, and the interval between them survives mostly through evidence found elsewhere. Rock is constantly being rewritten. Oceanic crust sinks back into Earth at subduction zones, carrying sediments and minerals into the interior. Heat and pressure transform buried rocks through metamorphism, changing their original textures and chemical signatures. Erosion strips mountains and plateaus away, carrying their fragments into younger landscapes or out to sea. Across hundreds of millions of years, these processes can remove entire chapters from the surface. The surviving record, therefore, has to be read indirectly. Ancient sediments contain isotope ratios of sulfur, carbon, molybdenum, and iron that preserve traces of the environments in which they formed. Volcanic rocks can retain magnetic signatures, recording the direction of Earth's magnetic field when their minerals cooled. Tiny fossils embedded in chert can survive where softer materials have been altered, preserving evidence of organisms that lived during otherwise poorly documented intervals. These clues rarely speak alone. A sulfur isotope pattern can reveal changes in ocean chemistry, while carbon isotopes can track shifts in the global carbon cycle. Molybdenum and iron can provide further evidence about oxygen levels and the chemical state of ancient seas. Paleomagnetic measurements help place those rocks on a changing planet, revealing whether sediments formed near the equator, at high latitudes, or somewhere between them. Each measurement narrows the range of possible histories. New methods continue to make the fragments more useful. Improved isotope geochemistry can detect chemical changes at finer scales. Radiometric dating can establish more precise ages for surviving rocks. Molecular clocks, built from genetic differences among living organisms, can estimate when major lineages may have diverged, even when their earliest fossils are missing. None of these methods restores the lost rocks, yet together they allow separate traces to be compared across continents and across immense spans of time. The boring billion is therefore known through a mosaic of evidence. Some pieces come from minerals, others from fossils, magnetic fields, chemical ratios, and genetic history. The picture remains incomplete, as every reconstruction of deep time must be, yet its outline becomes sharper whenever another surviving fragment is measured with greater precision. We are learning the history of a vanished world while also developing the instruments and methods capable of reading a record this old. Across the continents, certain rocks preserve a record written in bands of color. Banded iron formations formed in ancient seas, long before animals occupied the seafloor, and long before forests, reefs, or familiar ecosystems existed. Their alternating layers of iron-rich minerals and silica record changes in seawater chemistry and the availability of oxygen. Some became valuable sources of iron ore, yet their deeper importance lies in what they preserve. They are fragments of an ocean whose chemistry belonged to a world with no familiar complexity. The same rocks can also carry clues about the great freezes. During the snowball Earth intervals, dissolved iron could accumulate in ocean waters, as ordinary biological processes were greatly reduced. Hydrothermal activity continued supplying iron from Earth's interior, while oxygen became scarce in much of the ocean. When conditions changed, iron could precipitate and settle into layers. The resulting formations became geological markers of an ocean undergoing a profound chemical transition. Cap carbonates tell another part of the story. These limestone layers appear directly above glacial deposits in many regions, forming during the rapid climatic recovery that followed severe Neoproterozoic glaciations. Their unusual chemistry reflects intense weathering and a carbon-rich atmosphere interacting with newly exposed rock and seawater. They are like a thin mineral boundary between two different planetary states, one dominated by ice and the other by warmth and accelerated chemical exchange. Far away in Shark Bay, Western Australia, living stromatolites offer a different connection to deep time. Their layered structures are built by microbial communities that trap and bind sediment as they grow. Fossil stromatolites extend back more than 3 billion years, making this living tradition one of the oldest visible continuities in the history of life. Modern examples are not identical copies of their earliest ancestors, since environments and microbial communities have changed, yet the basic process can still produce structures resembling those preserved in ancient rock. The great unconformity in the Grand Canyon makes missing time tangible. Along the canyon walls, much younger rocks rest directly upon far older formations, with an immense interval absent between them. The boundary represents erosion and removal on a continental scale, followed by the deposition of younger sediments. Standing near such a contact means standing beside a surface that separates chapters of Earth history by hundreds of millions of years. In the Flinders Ranges of South Australia, another trace remains. Rocks associated with the Sturtian glaciation preserve glacial debris, including stones carried and released by moving ice in ancient marine environments. Those deposits formed at latitudes that were once much closer to the equator than the region occupies today. The hills now rise through an arid landscape, while their rocks retain evidence of an ocean once occupied by ice. None of these formations belongs only to a museum display. They are pieces of the physical earth that survived the immense recycling of continents, oceans, climates, and life. Banded iron, glacial deposits, cap carbonates, stromatolites, and ancient unconformities remain scattered across inhabited continents. Their surfaces can be touched, their layers can be measured, and their minerals can still be analyzed with instruments sensitive to chemical traces left by vanished oceans and climates. The vanished world is gone, yet parts of it remain embedded in stone, waiting for careful observation to turn those fragments into a readable history. A planet can spend 1 billion years preparing for complexity, while giving almost no outward sign that anything important is happening. That possibility changes the way we read distant worlds. What appears inactive from far away may be passing through a long interval of preparation, with chemistry, geology, and biology rearranging themselves on timescales too large for a human lifetime to witness. When astrobiologists examine planets around other stars, they often search for clues that stand out. An atmosphere containing gases that should react with one another can suggest continual biological or geological replenishment. Seasonal changes may reveal active climates, While unusual chemical patterns can point toward energetic processes, these signals are valuable, yet they describe activity at a particular moment. Earth offers another possibility, one in which the most important story can unfold while the visible surface gives very little away. During the boring billion, much of the machinery needed for complex life was being assembled inside conditions that would have seemed inhospitable. Ocean chemistry constrained nutrients. Oxygen remained scarce compared with modern conditions. Much of the land lacked the ecosystems familiar today. Across immense stretches of time, biological change could appear modest when viewed through the incomplete record preserved in stone. Still, cells were refining internal structures, populations were adapting to difficult chemical environments, and geological processes were altering the availability of essential elements. This creates a useful distinction between habitability and biological activity. A world can possess environments where life persists without producing forests, reefs, animals, or an atmosphere rich in obvious biological gases. Microorganisms can occupy narrow chemical niches and remain abundant enough to shape their surroundings, while larger forms of life remain absent. Such a planet might look uneventful through a distant telescope, even though its biosphere is influencing the chemistry around it. The lesson becomes especially important when scientists study exoplanets whose atmospheres appear stable and unremarkable. A planet with little seasonal variation, weak atmospheric disequilibrium, or modest biological signatures cannot automatically be placed among the sterile worlds. Its surface conditions may conceal activity occurring in oceans, sediments, ice, or mineral rich environments. Even a faint atmospheric signal can have multiple explanations, and the absence of a strong signal does not establish the absence of life. Earth also reminds us that evolutionary potential has its own history. Complex organisms require more than living cells. They depend on energy, nutrients, suitable chemistry, environmental stability, and enough time for biological innovations to accumulate. Those ingredients can emerge in stages, separated by hundreds of millions of years. A world may therefore spend an enormous portion of its existence becoming capable of something it has not yet produced. Seen this way, the boring billion becomes less a story about delay and more a lesson about hidden preparation. The universe may contain planets whose surfaces appear uneventful while their oceans, minerals, atmospheres, and microscopic inhabitants are steadily reshaping one another. Their greatest biological chapter may still lie far ahead, and from a distant point of observation, that future could be almost impossible to recognize. There is a deeper symmetry in this idea. Earth once occupied the kind of state that might have been dismissed from a distance, yet that same interval helped establish conditions from which complex ecosystems eventually emerged. Life does not always announce its future through spectacle. Sometimes the important work happens through accumulation, with small chemical changes preserved across immense stretches of time. There is a record of the boring billion that does not sit in a canyon, a mountain range, or a layer of ancient stone. It is carried within living cells, passed through generations, in structures that have remained part of complex life for more than 1 billion years. Each time a cell releases energy, an inheritance from that era is being used again. Inside, most of your cells are mitochondria, tiny structures that help turn food and oxygen into usable chemical energy. They are descendants of bacteria that entered into a lasting partnership with an ancestral complex cell. That partnership arose during deep time, probably more than 1 billion years ago, and it changed the possibilities available to eukaryotic life. The bacterium supplied an efficient way to process energy, while the host provided a protected environment. Neither lineage remained separate. Together, they became something new. Mitochondria still preserve clues to that origin. They contain their own small genomes, distinct from the much larger genome stored inside the cell nucleus. Their genetic machinery carries bacterial traces, while their surrounding membranes preserve clues to former independence. The arrangement is a biological fossil that continues functioning. It is ancient history operating inside a living organism. The inheritance becomes especially clear when scientists follow mitochondrial genetic material through families. In humans, mitochondria are generally passed from mother to child, allowing their genetic sequences to preserve lines of ancestry across many generations. Researchers can compare small changes accumulated through time and use them to reconstruct portions of human population history. Those studies reach back tens of thousands of years, while the mitochondrion itself carries a history measured on a scale more than 100 times longer. Plants preserve another branch of the same deep story. Chloroplasts, the structures that perform photosynthesis inside plant cells, descend from cyanobacteria that became permanent partners of ancestral eukaryotic cells. Their presence transformed the way those cells captured energy from sunlight. The green tissues of forests, grasses, mosses, and countless other plants therefore contain descendants of an ancient biological union. The partnership began long before forests existed, during an era when the planet was still reshaping the chemistry that complex life would eventually inherit. Even the oxygen entering your lungs connects to this older history. Cyanobacteria have been releasing oxygen through photosynthesis for billions of years. Their activity helped alter the chemistry of oceans and atmosphere long before animals appeared. The oxygen available to complex organisms today belongs to a planetary cycle stretching far beyond human history, carrying contributions from lineages that survived immense environmental change. There is something intimate about this inheritance. The boring billion can seem impossibly remote when measured through rocks and geological time, yet its consequences remain active in every ordinary moment of metabolism. The structures inside your cells are descendants of partnerships formed when Earth was still experimenting with complex cellular life. Their membranes, genomes, and energy pathways preserve evidence that ancient evolutionary events were not temporary episodes. They became permanent parts of biology. Your body, therefore, contains more than living tissue assembled in the present. It carries layers of biological history, folded into cells and renewed with every breath, every meal, and every moment of energy use. The distant world of the boring billion is no longer only something reconstructed from fossils and chemistry. Part of it is still working inside you. History often gathers around moments that can be named in a single breath: an asteroid strike, a volcanic eruption, a continent shifting beneath an ocean, a sudden advance of ice. Such events leave marks that can be traced across layers of rock, and those marks naturally become the landmarks of the story. The geological record itself encourages this habit. Catastrophes can bury organisms, fracture landscapes, alter minerals, and change the chemistry of oceans and atmospheres. Their effects may become unusually visible in stone. Long periods of gradual change are harder to preserve. A billion years of ordinary biological adjustment can leave fewer obvious signatures than one brief environmental upheaval. That imbalance makes the boring billion unusual. Some of the most consequential developments in the history of life unfolded across an era whose surviving evidence is scattered, altered, and difficult to interpret. The emergence of complex cells, the the reshaping of ocean chemistry, the gradual accumulation of oxygen, and the changing availability of nutrients were processes measured in immense stretches of time. Their importance was immense, even when their physical traces were faint. A cell becoming more complex does not create a mountain range. A chemical shift in an ocean does not carve a canyon in a single afternoon. Oxygen, accumulating through countless biological reactions, leaves no single moment that can be placed on a calendar. Each change becomes meaningful through accumulation, with one alteration influencing another until the character of the planet has been transformed. The same pattern appears in geology. Continents move by increments too small for any human observer to notice. Minerals circulate through rock, water, and living systems. Sediments gather grain by grain. Nutrients move between ocean floors and surface waters. These processes rarely offer a dramatic scene, yet together they continually alter the conditions available to life. This gives the long middle chapters of Earth history a different kind of importance. Catastrophes can redirect evolution, but they operate within environments that have already been shaped by countless, less visible processes. The conditions surrounding an extinction, a radiation of new organisms, or a major climate transition have histories of their own. Those histories may stretch backward through intervals that appear almost empty when viewed through the fossil record. The boring billion, therefore, changes what absence can mean. A sparse fossil record does not necessarily describe a world without meaningful biological activity. Limited, visible change does not imply that evolution has stopped. A stable landscape can conceal continual adjustment within cells, sediments, oceans, and minerals. For a planet observed from far away, this distinction becomes even more important. A distant world may show few dramatic atmospheric changes and no obvious surface transformation, while chemistry and biology continue operating across timescales beyond direct observation. What appears uneventful may represent a long period of preparation. Earth was shaped through these accumulated intervals as much as through its great disruptions. The planet became capable of complex life through processes that rarely announced themselves. Their traces survived in cells, minerals, sediments, and chemical signatures, waiting to be assembled into a larger story. The boring billion reminds us that a planet does not need to look busy to be changing. Some transformations require patience measured in geological time. They proceed through chemistry, biology, and geology, each contributing a small alteration that may eventually become the foundation for something entirely new. 1 billion years ago, Earth already possessed the essential ingredients of a living planet, yet those ingredients were arranged into a world almost unrecognizable from the one beneath our feet. The oceans were different, the atmosphere was different, and the continents occupied different positions. None of this made that earlier Earth incomplete. It was one stage in a planetary process that has never truly stopped. Even the atmosphere we now take for granted is temporary. Oxygen makes up about 21% of the air today, but that proportion has shifted repeatedly through geological history. During the Carboniferous, oxygen may have reached about 35%. Such enriched air helped support exceptionally large insects and other arthropods. During other intervals, oxygen fell sharply, placing greater demands on animals and contributing to environmental stress. The atmosphere has always been part of Earth's changing architecture. The continents reveal the same motion on a larger scale. North America and Europe continue to separate as the Atlantic Ocean widens, at a rate comparable to the growth of a human fingernail. Farther south and east, Africa is being pulled apart along the East African Rift, where the crust is gradually stretching and thinning. These movements are almost imperceptible within a human lifetime, yet given enough time, they redraw oceans, mountains, coastlines, and climates. Geologists expect this rearrangement to continue. In roughly 250 million years, the continents may gather into another supercontinent, often described as Pangea Ultima. The exact arrangement remains uncertain because plate motion can change, but the broader pattern is familiar. Continents assemble, fracture, drift, and assemble again. What appears permanent from a human viewpoint becomes temporary when measured against the age of the planet. The Sun also participates in this long transformation. Its energy output has been gradually increasing since the early history of the solar system. In roughly 1 to 2 billion years, the growing brightness is expected to push Earth's climate beyond the conditions that sustain today's oceans and complex surface ecosystems. Water loss and atmospheric changes would reshape the planet profoundly. Deep beneath the ocean floor, however, some forms of microbial life might endure around hydrothermal systems after surface environments become increasingly difficult for complex organisms. That distant future places the boring billion in a different frame. The world of that era was not waiting for Earth to become something more recognizable. It was already undergoing the same kind of planetary change that continues today. Oxygen shifted, continents moved, oceans evolved. Life altered its surroundings, while those surroundings shaped life in return. Coastlines will change, mountains will rise and erode, oceans will open and close, and the atmosphere will continue to respond to geology and biology. Earth has no final form waiting at the end of its history. The billion years that once seemed empty now fit into a broader pattern. A planet can spend immense stretches of time rearranging its foundations while appearing almost unchanged from a human perspective. Those intervals can shape everything that follows. Earth is still in motion, still exchanging matter and energy, still becoming something different. The same principle applies to life itself. Every generation inherits a world shaped by earlier generations, then alters that world in ways that become part of the conditions inherited by those that follow. Change is the background state of a living planet. At the south rim of the Grand Canyon, a person can stand beside a boundary separating rocks formed in different chapters of Earth's history. The Great Unconformity marks missing time, with younger Cambrian strata resting directly above much older rocks. In places, hundreds of millions of years of geological history are absent from the visible sequence. It is a reminder that the planet's story is written through gaps as much as surviving pages. That missing interval cannot be read as though every vanished year were preserved somewhere beneath the surface. Oceanic crust has been recycled into Earth's interior. Mountains have been worn down. Sediments have been buried, heated, folded, and transformed. Ancient surfaces have disappeared beneath newer ones. The boring billion survives through fragments, scattered across continents and locked inside minerals, sediments, fossils, and chemical signatures. Those fragments reach into the living world around us. The mitochondria inside human cells descend from an ancient partnership between an archaeal host and a bacterium, an event that occurred deep in the Precambrian around 1.5 billion years ago. Mitochondria still retain their own genetic material, preserving a trace of that former independence. Their descendants now help power every movement, heartbeat, and thought. Another inheritance surrounds us in every green landscape. Chloroplasts within plant cells descend from cyanobacteria that became permanent partners inside other cells. The oxygen entering our lungs today also belongs to a lineage that reaches back billions of years, carried forward by photosynthetic organisms whose activity helped reshape the atmosphere. The chemistry that once belonged to microbial worlds became part of the biological machinery of forests, animals, and people. Even the iron circulating through human blood belongs to a much older planetary cycle. Iron moved between oceans, sediments, minerals, and living systems for immense spans of time. Some chemical pathways shaping those cycles were already operating before animals existed. A metal passing through a blood vessel today, therefore, participates in a history far older than the body carrying it. The scale can be difficult to hold. A human life measures decades. A civilization measures centuries or millennia. A billion years stretches across so many generations that lives become almost impossible to separate from the interval. Geological change works through repetition, accumulation, burial, uplift, erosion, and chemical exchange, each process leaving traces that may survive long after the organisms themselves have vanished. That is why the boring billion matters beyond its place in a timeline. It changes the meaning of apparent stillness. A landscape can seem unchanged while its chemistry is shifting. An ocean can remain hostile to animals while microbial communities alter the resources available to future life. A planet can appear biologically uneventful, while its deepest systems prepare conditions that eventually support forests, animals, and intelligence. Earth spent a billion years becoming more than a surface that could host life. It became a world whose chemistry, geology, atmosphere, and biology could sustain increasingly complex forms of existence. The great lesson is measured less by spectacle than by duration. What looks like an empty interval from a human perspective can contain the construction of an entire living world. The billion years when Earth seemed unfamiliar were still part of the same continuous story. The rocks remember fragments of them. Our cells carry older traces. The air itself bears their inheritance. Beneath every modern landscape, the unfinished history of the planet continues to shape the world we call home.